Okay, so we've been talking about cell membrane. Cells have a membrane so that they can keep things in, but they're supposed to be inside the cell and keep other things out. But obviously there has to be many times transport for molecules to get in and out. And that little video kind of went through that. You might want to look at that again on your own. But in general, the most common form doesn't require any of those proteins. Transport proteins like here. Molecules like water or oxygen or CO2, which have no charge, they're not polar, they can actually cross the plasma membrane because these are actually, the plasma membrane has a phospholipid bilayer, and the outside here and inside here are polar. Okay, so they have a charge. So anything with a charge will not be able to get through. So here's diffusion again. So we've got a glass of water, and we've got some dye in it. You put the dye in, and eventually it diffuses throughout the whole volume of water so that the concentration is equal. That's what diffusion does, it equalizes concentrations. So if you have a higher concentration on one side, it diffuses to the other side until both sides are equal. So osmosis is water molecules moving by simple diffusion. Osmosis is important. Well, most of our cells, all of our cells are basically water, 70%. So, and water is the universal solvent. So when, in terms of water and the concentration, when the water molecules are equal in concentration, that's an isotonic solution in terms of the solute. Cells that are surrounded with a solute of higher concentration than the cell's interior are called hypertonic. So it's going to flow into the cell versus when the concentration is higher inside the cell, then it will go, it's hypotonic, and will go outside. You can see that here. So we've got this semi-permeable membrane, and the water molecules move that way. And when it's hypotonic, let's see, apparent. Oh, this is showing isotonic. Okay. So another way that things move are in vesicles, which are little spheres that might carry some substance, whether it's ions or neurotransmitters. So here's a little vesicle, and it's got things inside, and it might want to, let's say the vesicle has things inside that it wants to put into the bloodstream. So it will contact the plasma membrane, and then it would open up and these particles will then flow out. Or, as another example, which we'll learn about, this is a neuron. Another neuron. These are vesicles with neurotransmitters. They'll come to the edge and then release the neurotransmitters into the synapse. And then there's receptors all along here that they dock onto. Okay, so in that case, the vesicles are actually carrying neurotransmitters and then releasing them into the synapse. So in that case, in the case of let's say a cell doing exocytosis, releasing stuff out of the cell, and then endocytosis inward. So, and then receptor-mediated endocytosis, there are receptors that recognize and attach to specific substances, and includes phagocytosis, nonspecific endocytosis of large substances, and pinocytosis. You're not going to have to, by the way, memorize this, okay? Just the main thing you have to know about this sort of thing is you have to understand diffusion and active and passive transport. And I might say, what's cytosis? One of them, and you'll have to answer. Here's an example of exocytosis. These are vesicles. This is the plasma membrane, and it's releasing, let's say, waste products into the bloodstream. Endocytosis, this is a white blood cell, part of the immune system. So if it comes across, let's say, some microbe that's not part of where it's supposed to be, it will engulf it, and that's called endocytosis. It'll take it inside the cell where it'll be broken down. Like this. That way. Okay. Okay. Now, in the past, let's say 30 or 40 years ago, it was thought that there were 5 basic kingdoms of life. Monera was microbes, like bacteria that didn't have cells that didn't have a nucleus. There were protists, eukaryotic cells that did have a nucleus, but single-celled. And then there were plants, animal, and fungi. Okay? That's all gone. We now know, this is what this diagram is called a phylogenetic tree. You can read it from old to young. So at the very bottom, we have a most recent common ancestor right here. Now, and then eventually things split. And you had one group that went off to this way, and that's bacteria. And then there was this other group that went this way. And then this other group then split again later in time. And those were archaea and finally eukaryote. Okay. And anytime you see a place where two things split, say right here, that means both those lineages had a most recent common ancestor. Okay? And then later there was another split. So these are called nodes. And you can see nodes all over the place. Okay. So now it's thought that there are three domains of life. There's a couple different hypotheses about whether eukarya split from an archaea lineage or whether archaea and eukarya split prior to that. But one thing we can say when we look at this is that wherever there's a node, the two lineages are more closely related to each other than another node. So even though bacteria and archaea are often referred to as prokaryotes. No nucleus. Okay. It turns out, even though archaea and bacteria both have no nucleus or any organelles, okay, they are actually not as closely related to each other as archaea and eukarya are, even though eukaryotes do have a nucleus and organelles. Okay. And we know this because having a nucleus or not having a nucleus is just one trait. And it turns out there's a whole bunch of things that make archaea and eukarya more related to each other than either eukaryotes or archaea are to bacteria. Okay? They use more molecules in common. They share more genes in common, all kinds of things. Oh, one more thing. So this diagram basically says that all life is related, that all life is descended from a common ancestor. Happened a long, long time ago. Down here. the reason it's thought that is that all life here that we see up on top, whether it's bacteria, archaea, or eukaryote, they share certain things in common. So, what things might you suppose they share in common? In terms of, let's say, the molecule of inheritance. What's the molecule of inheritance that most of you know? Starts with a D? DNA. Okay. So all life uses DNA and uses the same exact genetic code for instructions to make proteins. And they all use RNA as a mediator to translate that into proteins. So now, if you had multiple origins of life, would you expect the same molecule of inheritance? Like, is there something about DNA that's magic? Anybody? Yes? No? Yes? DNA is magic. It is. No. If we had another form of life, it's likely that it would use a different, just by random chance, a different molecule of inheritance that could store that kind of information. And it might use a different genetic code to do that in terms of turning those things into amino acids. So what this tells us actually is that in terms of life on Earth, it looks like it only originated once, because all life on Earth shares this genetic code. And there might have been other life forms that originated, but they're not here anymore. Maybe they got outcompeted. And maybe there are fossil evidence of that, but we don't really see it. Or the third hypothesis, alternate hypothesis, is that maybe there are other forms of life on Earth, we just don't recognize it as such. So this goes back to the scientific method. So a lot of times you can come up with all alternate hypotheses, and then what you want to do is each one makes specific predictions. And right now, the one that makes the predictions that we actually see in nature is the one that origin of life only once on this planet. Okay. So a prokaryote is a single cell organism that lacks a nucleus and organelles, and that includes both bacteria and arc and then eukaryotes that do have a nucleus. And organelles. And organelles are, an example is mitochondria. All cells have mitochondria. Mitochondria are basically the power plants of cells. And most cells have thousands and thousands of mitochondria. First of all, okay, so up here we have a bacteria. Inside is the DNA. There's still a plasma membrane, right? There's a cell wall. There are these ribosomes that they use to turn the instructions from DNA into amino acids, which form a protein. And then there's the cytoplasm. Okay, and they don't have mitochondria. And they also, a lot of bacteria have a flagellum. It seems like the oldest ones have a flagellum, but not all of them do. And then a eukaryotic cell is a lot different, is a different animal entirely. It's got a nucleus, and the nucleus is where the DNA is. And the DNA, all it is instructions about how to make a protein. That's it. It doesn't actually make a protein, it's just the instructions. And it transcribes those instructions into something called RNA, which we'll learn about later. And then that RNA is taken out of the nucleus. And let's see. Oh. And then there's ribosomes right here that actually dock with the RNA and then actually create proteins. And then there's all this other stuff that's going on. You don't have to really know much about it. Rough ER, smooth ER, Golgi apparatus, and all that. Most of it has to do with modifying the protein after it's made. Oh, and then here's a mitochondria. And the mitochondria, usually there's thousands in each cell, depending on how much energy the cell needs. And the mitochondria provide the energy source of the cell, which is called ATP, which we'll learn about. And ATP is made from glucose. Okay. And then there's other things that are going on. This is a plant cell. And plant cells also have another organelle called chloroplast, where photosynthesis actually takes place. Okay, so animal cells do not have a cell wall. They have a cell membrane, but not a wall. Plant cells have a cell wall, and also fungi have a cell wall. So a number of eukaryotes have cell walls and others don't. Bacteria also have cell walls, some do. And these are the different functions. And you can go over that yourself. But in general, the nucleus for a eukaryotic cell, what's in the nucleus of a eukaryotic cell? Anybody? DNA. Okay. And then there's a nuclear envelope, nuclear pores, so that things can move out of the nucleus into the cytoplasm. And then all these things, I'm not gonna go over that in detail. Lysosomes are basically, in animal cells, they act as a garbage dump. Or a lot of things that are made, let's say proteins, are actually that after they're used, they're broken down into the constituent parts. So a protein is made up of amino acids, so the lysosome will break it down into amino acids to be reused. Mitochondria are the power plants of cells. Cells that need a lot of energy, like heart cells or muscle cells, have thousands. Others that don't, like fat cells, don't have very many mitochondria. And they produce the energy that a cell used in the form of what's called ATP. Adenosine triphosphate. And ATP is made from glucose metabolism. So our bodies use glucose. They're taken in. You need insulin to burn up glucose, and then glucose is turned into ATP through cellular respiration, which we'll learn about next. There's the cytoskeleton, so unlike a prokaryote like an archaea or bacteria, eukaryotic cells have this cytoskeleton that kind of gives it shape and helps things move around. Plant cells and some algae have chloroplasts, and chloroplasts are made up of these things called thylakoid membranes that are part of photosynthesis, and we'll go over that also.